Hello everyone, welcome to my presentation. My name is Wenye Liu and I'm a postdoctor of Liu Jinxia's research group at Huazhong Agricultural University. I'm pleased to introduce our work published in Cell Regeneration about zebrafish F1 and 2 modulate erythropoiesis via regulating GIT1A expression and Wnt signaling to facilitate hypoxia tolerance. Thank you for your time, and I hope you enjoy it. Firstly, I would like to introduce the background of our study. In our lab, the hematopoietic development in zebrafish is one of the main research directions. In this study, we mainly focus on zebrafish erythropoiesis. Zebrafish erythropoiesis comprise two successive waves, the primitive wave and the definitive wave. The primitive wave generates erythrocyte, granulocyte, and monocyte for the development of early embryos, while the definitive wave generates definitive HCC to maintain blood cell production throughout the zebrafish lifetime. A sophisticated network of lineage-specific transcription factors, SCL-GIT1 and MO2 and so on, was shown to function in erythrogenesis while regulating the expression of erythropoietic genes. Previous studies in our lab have found that F1 and 2 can act as transcriptional suppressors to inhibit the TGF-beta signaling pathway and Wnt beta-catenin signaling during early zebrafish embryogenesis. F2 has been reported to protect cells against hypoxia-induced cell death. However, the use of F1 and F2 in hypoxia tolerance by regulating erythropoiesis and the related underlying mechanisms remain almost entirely unclear. So, I next would like to introduce our findings in this article published in Cell Regeneration. In this study, we knocked out F1 and F2 in zebrafish to generate homologous mutants by CRISPR-Cas9 system, and F1 or F2 mutants exhibit almost no F1 protein and F2 protein. Next, we examine the stress resistance of the mutants to hypoxia infection. We found that F1 and F2 mutants exhibit increased sensitivity to hypoxia in both embryo, larvae, and adult fish. Accompanied with the downregulated HIF1A protein level and the decreased expression of hypoxia-inducible genes in the mutants under hypoxic conditions. Under hypoxia stress, fish tend to adapt it by increasing the number of red blood cells and promoting the oxygen-carrying capability of hemoglobin. So we measure the red blood cells of the mutants by odeoneating staining, and the results show that the mutants have fewer odeoneating positive cells. With similar results, the expression of erythrocyte markers HBE1, BBE2, and HBE3 were significantly downregulated in the mutants by which analysis. Meanwhile, knockdown of F1 and F2 caused a significant reduction in the number of DLL positive cells and GIT1A positive cells, and caused decreased expression of GIT1A and HB3 in both DLL positive and GIT1A positive cells. These results indicate that F1 and F2 are required for erythropoiesis. As we mentioned above, SAL, MO2, and GIT1A are essential for zebrafish erythropoiesis. In this study, we found that in F1 and F2 mutants at 14 and 24 HPF, GIT1A expression was clearly reduced, and at 14 HPF, the mutants showed significant upregulation in the expression of both SAL and MO2, in contrast to significant reduction in the expression at 24 HPF. F1 and F2 were able to regulate the anterior-posterior pattern of the zebrafish axis by modulating Wnt beta-catenin signaling. In this study, we examine the expression of Wnt signaling ligands Wnt3 and Wnt16, Wnt activated receptor FZD2, and Wnt beta-catenin signaling target gene axin2 in AF1 and F2 mutants. At 16 HPF, axin2, Wnt16, and FZD2 all showed an increased expression in the mutants, and at 24 HPF, Wnt16 and FZD2 maintained upregulated expression, while axin2 were obviously downregulated in the expression in the mutants. Consistent with tendency of beta-catenin and phospho-beta-catenin protein levels in F1 and F2 mutants, knockdown of either F1 or F2 also results in the decrease of GAT1A positive cells, with a decrease in beta-catenin level at 24 HPF. Next, we examine the Wnt beta-catenin activities in the mutants using top flash luciferase proteases, and observed increased top flash luciferase activities at 16 HPF, while reduced activities at 24 HPF in the mutants. Besides, F1 and F2 mutants showed reduction in the binding enrichment of TCF4 in both SCL and MO2 promoters at 24 HPF. This result indicates that F1 and F2 may regulate SCL and MO2 transcription by modulating Wnt beta-catenin signaling in a developmental stage specific manner. Studies have identified that F1 and F2 are involved in epigenetic modification, so we test the levels of epigenetic modified protein. We found that F1 and F2 mutants showed a significant reduction in the protein level of H3K27AC, H3K4ME1, and H3K4ME3, in contrast to upregulation in the protein level of H3K27ME3 at both 14 HPF and 24 HPF. Meanwhile, knockdown of either F1 or F2 led to decreased number of both DLL positive and GAT1A positive cells, accompanied by increased H3K27ME3 level. H3K27ME3 protein level and the expression of GAT1A and HBB3 could be recovered by H3K27 methylation inhibitor in F1 and F2 mutants. In addition, typical PCR analysis shows that the binding enrichment of H3K27ME3 was enriched in GAT1A promoter, in contrast to no obvious changes in the H3K27ME3 enrichment in both SCL and MO2 promoters in the mutants. In summary, we found that F1 or F2 dysfunction caused reduced red blood cells and hypoxia tolerance in zebrafish, and loss of 1 and F2 caused significant changes in the expression of epigenetic modified histones, with a significant increase of H3K27ME3 enrichment in GAT1 promoter. Meanwhile, deficiency of F1 or F2 result in dynamic expression of Wnt beta-catenin signaling during erythropoiesis, with reduced beta-catenin level and enrichment of TCF4 in both SCL and MO2 promoters. We are grateful to Professor Meng Anming and Professor Jing Lili for providing transgenic lines, and thank fund agencies for funding spots. That's all for my presentation. Thank you very much.